Chapter 10, Section 2, Applying Mendel's Principles. Learning objectives: explain how probability can be used to predict inherited traits. Explain how alleles segregate when more than one gene is involved. Identify Mendel's contributions to our understanding of genetics. Nothing in life is certain. There's a great deal of wisdom in that old saying and genetics is a fine example. If a parent carries two different alleles for a certain gene, we can be sure which of those alleles will be inherited by any one of the parent's offspring. However, think carefully about the nature of inheritance, and you'll see that even if we can't predict the exact future, we can do something almost as useful. We can figure out the odds. Whenever Mendel performed a cross with pea plants, he carefully categorized and counted the offspring. Consequently, he had plenty of data to analyze. Upon analyzing his data, Mendel realized that the principles of probability could be used to explain the results of his genetic crosses. Probability is the likelihood that a particular event will occur. As an example, consider an ordinary event, such as flipping a coin. There are two possible outcomes of this event. The coin may land either heads up or tails up. The chance or probability of either outcome is equal. Therefore, the probability that a single coin flip will land heads up is 1 chance in 2. This amounts to half, or 50%. If you flip a coin 3 times in a row, what is the probability that it will land heads up every time? Each coin flip in an independent event with a half probability of landing heads up. Therefore, the probability of flipping 3 heads in a row is half times half times half equals 1/8. The way in which alleles segregate during gamete formation is every bit as random as a coin flip. Therefore, the principle of probability can be used to predict the outcomes of a genetic cross. Like a coin toss, each F2 gamete has a 1 in 2 or half chance of carrying a small g allele. There are 2 gametes, so the probability of both gametes carrying the little g allele is half times half, or 1/4. In other words, roughly 1/4 of the F2 offspring would have yellow pods, and the remaining 3/4 should have green pods. The predicted ratio: 3 offspring exhibit the dominant trait and 1 offspring will exhibit the recessive trait, showed up consistently in Mendel's experiments. For each of his 7 crosses, about 3/4 of the plants showed the trait controlled by the dominant allele. About 1/4 showed the trait controlled by the recessive allele. Segregation did occur according to Mendel's model. As you can see in the F2 generation, not all organisms with the same characteristics have the same alleles. Both the big G and big G allele combinations resulted in green pods. Organisms that have 2 identical alleles for a particular gene, or big G, or little g in this example are called homozygous. Organisms that have 2 different alleles for the same gene, such as big G, are called heterozygous. Probabilities predict the average outcomes of a large number of events. One of Mendel's most revolutionary insights followed directly from his observation of F1 crosses. Every organism has a genetic makeup as well as a set of observable characteristics. All of the pea plants with green pods had the same phenotype, or physical characteristic. They did not, however, have the same genotype, or genetic makeup. One of the best ways to predict the outcome of a genetic cross is to do a diagram known as a Punnett square. A Punnett square is a mathematical model used to predict the probability of the genotype and phenotype of combinations of a genetic cross. Constructing a Punnett square is fairly easy. You begin with a square, then follow the principle of segregation. All possible combinations of allele in the gametes produce one parent, and they are written along the top edge of the square. The other parent of alleles are then segregated along the left edge. Next, every possible genotype is written inside the boxes within the square.
That face that is usually covered by our clothing has finally made an appearance.
Thanks for stopping by. Today we're talking about some basics of Punnett squares. Punnett squares measure the probability or likelihood of a genetic outcome based on a specific genetic cross. It doesn't tell you what will be, only what could be. For the simplest of Punnett squares, we'll cross two genotypes. These parent genotypes will be the same gene that may or may not contain different alleles. Let's look at an example of a genetic cross for eye color. Green eyes being dominant and blue eyes being recessive. We use big G, little g for the maternal genotype and little g for the paternal. So this is our cross. Mother has green eyes and father has blue eyes. We want to see the probability that their offspring will have either green or blue eyes. For this cross, we'll draw a square split into four quarters, a simple four-panel grid. This is the Punnett square. One of the parental genotypes will go on top, and the other will go on the side. It doesn't matter which genotype goes where, and the order of the letters really doesn't matter, but we conventionally write the dominant allele first for this fact. So your setup should look like this. Then you simply carry the letters through to their respective quadrants and match them up, like so. Each quadrant will represent a 25% chance that genotype will result in the offspring. So in this case, we have only two possible genotypes, big G, little g, and little g, little g. And each genotype has a 50% chance of showing up in the offspring. Similarly, you have two possible phenotypes, green eyes and blue eyes, with 50% chance of each. Let's look at another example. Cross two genes that are heterozygous for eye color in this example. So big G, little g crossed with big G, little g. These colors are just for illustration purposes. So we begin, we draw our Punnett square, and put one gene on top and the other down the side. And then carry letters through to their respective quadrants and analyze the results. This cross results in a 1 to 2 to 1 genotype ratio, with 25% homozygous dominant, or big G, 50% heterozygous, or big G, little g, and 25% homozygous recessive, or little g. Phenotypic ratio would be 3 to 1 ratio, with 75% green eyes and 25% blue eyes. Hopefully that helps you understand simple Punnett squares a little better. Genetic crosses can be much more complicated, and I'll make a video sometime in the future.
After showing that alleles segregate during the formation of gametes, Mendel wondered if the segregation of one pair of alleles affects another pair. For example, does the gene that determines the shape of a seed affect the gene of a seed color? To find out, Mendel followed two different genes as they passed from one generation to the next. Because it involves two different genes, Mendel's experiment is known as two-factor or dihybrid crosses. Single gene crosses are monohybrid crosses. First, Mendel crossed two breeding plants that produced only round yellow peas with plants that produce only wrinkled green peas. The round yellow peas had a genotype big R, big R, big Y, and the wrinkled green peas had a genotype little r, little r, little y, little y. All of the F1 offspring produced round yellow peas. These results showed that the alleles for yellow and round peas are dominant. The genotype in each of these F1 plants is big R, big Y, little y. In other words, the F1 plants were all heterozygous for both seed shape and seed color. You can see more about this on page 294 in your textbook. Next, Mendel crossed the F1 plants to produce F2 offspring. Remember that each F1 plant was formed by the fusion of a gamete carrying the dominant big R, big Y allele with another gamete carrying the recessive little r, little y allele. Would the two dominant alleles always stay together, or would they segregate independently so that any combination of alleles was possible? In Mendel's experiment, the F2 plants produced 556 seeds. Mendel noted that 315 of the seeds were round and yellow, while another 32 seeds were wrinkled and green. The two parental phenotypes, however, 209 seeds had combinations of phenotypes and therefore combinations of alleles that were not found in either parent. This clearly meant that the alleles for seed shape segregated independently from those of the seed color. Put another way, genes that segregated independently, such as the genes for seed shape and seed color in pea plants, did not influence each other's inheritance. Mendel's experiments results were very close to a 9 to 3 to 3 to 1 ratio that the Punnett square shown here on this page predicts. Mendel had discovered the principle of independent assortment. The principle of independent assortment states that genes for different traits can segregate independently during the formation of gametes. Independent assortment helps account for the many genetic variations observed in plant, animals, and other organisms, even when they have the same parents. Mendel's principles of segregation and independent assortment can be observed through 1- and 2-factor crosses. Mendel's principles of heredity, observed through patterns of inheritance, form the basis of modern genetics. These principles of heredity are as follows. The inheritance of biological characteristics is determined by the individual units called genes, which are passed from parents to offsprings. Where 2 or more forms, alleles of the genes for a single trait exist, some alleles may be dominant and others may be recessive. In most sexually reproducing organisms, each adult has 2 copies of each gene, one from each parent. These genes segregate from each other when gametes are formed. Alleles from different genes usually segregate independently from each other. Mendel's principles don't apply only to plants. At the beginning of the 1900s, the American geneticist Thomas Hunt Morgan wanted to use model organisms of another kind to advance the study of genetics. He decided to work on a tiny insect that kept showing up uninvited in his laboratory. The insect was the common fruit fly, Drosophila.